So this is the second part of the virus lecture. I've never used this software before, so I decided to try the little webcam feature in the corner. See how that works. You can always stare at our beautiful portrait of the Breakfast Club kids. If you get bored, perhaps one of the cats will visit at some point. Anyway, um, we talked about how the virus can get, come on, virus can get an envelope from the external membrane of a cell. You can also get it from the internal membrane system of a cell. So we have the endoplasmic reticulum here, we have the Golgi body here, and depending on where the virus is made, it will get its membrane in different places. And I keep saying membrane, but what I really mean is it will get its envelope. So for example, these ones here that are being made in the endoplasmic reticulum, they're going to bud off and have an envelope made of the membrane from there. And the same with the ones that are in the Golgi. They're going to bud off and they've got their membrane from the Golgi. All right, let's talk about the coronavirus specifically. This is a picture of the actual coronaviruses infecting a cell. These dark little dots are the viruses. So what they did was scientists took some cells and they put them in a petri dish and they put some virus on it. They inoculated it with the virus. This is a general drawing of the virus. It's got a spherical shape. The genome is a single strand of DNA, or RNA. It's not DNA, it's RNA. It has an envelope, and it's made in the Golgi body and the endoplasmic reticulum. And I want you to take note of these spike glycoproteins. And whenever I refer to glycoproteins, what I actually am talking about is, hold on, cats. Okay, what I was trying to say is that a spike glycoprotein is the same thing as a surface antigen. So those things mean the same. Oh my God. All right, there's thousands of different types of viruses, and they're classified based on their genetic material, whether it's DNA or RNA, whether it's single-stranded or double-stranded, and their host strains. And the SARS-CoV-2 virus has a single-stranded RNA. Ooh, this one is really cool. Okay. This virus is called a bacteriophage. It's a virus that infects bacteria. There's a lot of different kinds of bacteriophages, but they mostly look the same. This is what they actually look like under a microscope. Over here is a drawing. The capsid is this sort of hexagonal or whatever shape. Icosahedral? I don't know. Capsid is this thing over here. We've got the DNA inside the capsid. And what happens is there's this sheath, this hollow sheath, and the virus comes and sits down on the surface of the bacterium and just compresses itself and squirts the DNA down into the cell. And you can see that happening over here with some of the little DNA. It's blue. All right, the mechanism of action, how a virus works. There's a lot of different variations. The one we're going to do is for an animal RNA virus. It's the example in your book, I think, chapter 10. But it's not the same as the coronavirus, even though the coronavirus is also an animal RNA virus. So go ahead and watch this animation about how the coronavirus works and then come back. Also, watch this animation and then come back. All right, so in this image, you see a virus, and this virus has an envelope, and it also, here they're calling it the membranous envelope. There's also a capsid, they're calling it the protein coat here. You can see that it's made of the individual capsomeres. Remember, capsomeres are the units that make up the capsid. There's the glycoprotein spikes, which are the surface antigens. And then inside is the genome, which is RNA. So the first step is entering the cell, and the spikes on the surface of the virus bind to receptor proteins on the surface of the host cell. And this is how the virus recognizes its host cell. The shape of the glycoprotein spike and the shape of the cell receptor have to fit perfectly to each other in order to trigger entry into the cell. Here's how the coronavirus does it. So there's thousands of pre-existing coronaviruses. They live in wild animal populations. They're very similar to human viruses, and a lot of them can enter human cells without mutating. They just use the existing host cell receptors. This is the SARS-CoV-2 spike protein, the surface glycoprotein. This is the human cell receptor. It's called ACE2. So the ACE2 would be this thing on the surface of the human cell. In the case of the SARS-CoV-2 virus, it would be a mucosal cell probably that's lining the inside of the throat, or a cell in the lungs. And then the spike protein is right here. And you see they have to fit perfectly in order to trigger entry into the cell. All right, so we got that. Some viruses are highly specific and can only enter a very few cells, and some of them can infect a broader range of cells. Oh, and the ability of the viral glycoproteins to fit with the different surface cell receptors determines the host range, which, remember, is the type of cells that the virus can infect. For example, well, we have HIV, and then there's also a cat equivalent called FIV, feline immunodeficiency virus. The FIV will never be able to have an effect on humans because its surface glycoproteins don't fit any of the cell receptors in the human body on any of the cells. And it's the same with HIV for cats. The HIV glycoprotein spikes don't fit any of the cell surface receptors in cats. Also, I want you to note that as the virus is entering, its envelope is fusing with the host cell membrane, and it's going to leave its glycoproteins behind the membrane. All right, so then we have uncoating. The capsid comes apart, capsomeres go everywhere, and the viral genome comes out. The next step is the RNA synthesis by a viral enzyme. The virus uses one of its own enzymes. Sometimes viruses use enzymes that are already existing in the cell, and this is, one way in which the life cycles differ. Remember, an enzyme is a functional protein, just a protein that does things. So in this case, this virus is using its own pre-existing enzyme to synthesize RNA. And what's going to happen with that RNA is two things. First of all, the RNA is just going to be used, more and more and more and more copies are going to be made to put into the new viruses. But also, it could be used to create the proteins that will surround the new viruses, the new capsid. So protein and RNA synthesis, this is what we're going to learn about later in chapter 10. We have mRNA, which is a kind of RNA, and that's the kind of RNA from which the new viral proteins come. And then over here you can see the new RNA being made, copies and copies that will go inside the new viruses. Next is assembly. The capsomeres come and assemble all around the viral RNA, and then in this case the virus is going to bud out of the host cell, and it's going to take part of the host cell membrane with it, and as it goes, the surface glycoproteins assemble on the envelope. It becomes the viral envelope, and then it leaves, and thousands of viruses can be made per cell. Sometimes viruses are latent. They integrate their genetic material into the host chromosome, and then they lie dormant. This is an excellent life strategy for viruses because their DNA can be copied into every single descendant cell. And then, there's all these cells that have viral DNA in them without showing any effect at all. And then they wake up, or they activate at a later time. That usually is based on some external condition. It's different for every virus, and the mechanism is poorly understood. But herpes virus is one like this. There's also oncoviruses like this, which convert normal cells into cancer cells. All right, let's talk about vaccines and autism for a minute. There is no verified evidence that vaccines cause autism. So here's where that little wives' tale came from. In 1998, this guy described 8 children only, who showed what he thought were signs of autism within several days of being inoculated with a vaccine. That's way too soon to decide if a vaccine has caused something. No other scientists were able to replicate his results. He conducted his studies without approval from his hospital. He paid the kids at his son's birthday party to draw their blood. This is starting to sound like some kind of twisted horror film. Oh my God. Finally, his medical license was revoked, but by that point, the damage had been done. Whew. Also, vaccines don't contain mercury. I just wanted to dispel that misconception that often happens. All right, let's talk about vaccines and how they work. First, let's talk a little bit about the immune system. The immune cells include white blood cells. There's other different types of immune cells as well. But when the white blood cells encounter invaders, they recognize the surface proteins, the antigens, of the invader, of the pathogen. And there's a there for you. The point of the antibodies is to bind to the antigens on the surface of the pathogen and block them from binding with the surface receptors of the host cells. Then the white blood cells come and they digest the pathogens. Go ahead and watch this video and then come back when you're done. Okay. Vaccines, immune cells produce specific antibodies to defend against each antigen that they encounter, as you saw from the video. And the shape of the antibody has to fit the shape of the antigen perfectly. It takes a few days to find the right DNA to make the right antibody for each antigen. But immune cells can remember the antigens, and they store the DNA that creates that specific antibody so that the next time they see the antigen, they don't have to go through their entire library of DNA trying to find the right DNA that will create an antibody that fits whatever antigen they see at the moment. This allows an immediate response. Vaccines capitalize on this by introducing a small amount of weak or dead antigen in the body, and the immune cells can encounter the antigen, they can remember it, and they can reproduce an immediate response. So this graph shows the primary immune response. Well, first of all, there's concentration of antibody on one axis, and there's time on the other axis. The primary immune response is what happens with the vaccine. So initial exposure, there's not a lot of virus, so not a lot of antibodies have to be made. And then the second spike is when the body encounters the actual virus. And you can see that the amount of antibodies increases much more quickly, and it increases to a greater rate the second time that it sees the virus. Go ahead and watch this and come back when you're done. That's the end. Okay, there's one more thing that I want to tell you. Well, two more things. First thing is that people who are exposed to a lot of mild pathogens develop a lot of different antibodies, which is why they're resistant to different things that go around. Like preschool teachers, for example, don't often get sick because they are around like mild pathogens all the time. Okay. The second thing is that not everyone has the DNA that's necessary to make every antibody possible against every possible antigen. This is one reason genetic diversity is so very important. There's a wide array of what's called MHC genes, major histocompatibility complex genes, and those are what code for the antibodies. And so the wider range, the more difference in them that you have, the better your immune system is going to be. And there was a study where I have like 20 seconds. These men exercised in a t-shirt. They put the t-shirts in boxes. The women smelled them and they ranked them on how well they liked it. Then they tested the genes, and the ones who were most attracted to each other had the most different MHC genes, and that would give their offspring the best opportunity for survival.